Well, first of all, thank you so much for inviting me. My name is Jillis Grubben. I'm from the Wageningen University. I work at the plant breeding department, and today I would like to talk about the CRISPR-Val movement, which is not yet a startup company, but it is a project, a Dutch project, and we focus on a novel Cas enzyme, which should provide accessible genome editing using this enzyme called thermocas9. Just a little bit about the contents of the presentation then. First of all, I would like to present, okay, what is our CRISPR-Val movement? Would like to talk a little bit with you about the tech. So from the discovery of this enzyme all the way to breeding relevant applications. And also finally about some unique properties of the thermocas9 enzyme. So first of all, we have a global challenge, as you all know, where we see increasing climate variability. So we need more resilient and stable production systems. And another example is that we would like to all further reduce pesticide use overall. And that means also that we need more resistant crop varieties. And this all is not something that a small group of people wants. There's a broad coalition of food and feed associations that would like to support this. So what we believe is that the new genomic techniques, they really provide an opportunity here. They're not the silver bullet, but definitely they can help and accelerate breeding by offering better speed and precision compared to the conventional systems. Also in Europe, indeed, as Claudia already mentioned, we see that there are shifting EU perspectives on the new genomic techniques, and these indeed drive excellent market forecasts as well. So I'd like to quickly show you a graph here of the global market size of these breeding tools. So currently 2026, the total market is estimated at 10 billion. And I would like to focus on NGTs here. So the green bar, there we see that roughly 15% currently is made up from NGTs. And the forecast for 2036 would be that that would grow to a third. And then in 2046, it would even grow to almost half of the entire market size. So indeed, there seems to be really an excellent market forecast there. Focusing on the Netherlands, we see that in the Netherlands, we have a world-class plant breeding system. But based on a report that was done for the Dutch government, it was concluded that we really should invest and remove regulatory barriers to really stay ahead in the game. So while the potential is clear in the Netherlands and EU, mainly we see that adoption is relatively slow. So we asked the question, okay, what is the reason for this? So what we see is that the CRISPR-Cas9 licenses that are currently being offered by, for example, Broad and CVC are extremely expensive, especially for smaller companies. It's a very complex IP landscape, and these legal uncertainties, they really do limit the GEUs. And smaller breeders, they are in fact locked out because if you have to provide a percentage of your profit margin to, for example, to Broad and the CVC, if you would have a CRISPR-Cas license, which is the case, then you cannot make profit as a smaller company. So the first movers and the large companies, they really have a large advantage over the smaller companies that stay behind. Just to show that here in a graph, we have the Bayers and the Cortevas that are really advanced, and then the early adopters that have their own tech. And we see that as we also saw from the previous presentation, that in the Americas there's a big adaptation already, and Europe is still lacking behind. And we would like to really, in the future, see this a bit differently. These barriers that I mentioned, they really limit agricultural innovation currently. So then what we also see is that we would like to all feed the world. So we think that innovation should really be accessible to all breeders. And therefore, we should really boost biotech. And what I would like to show here, let me move around something. Okay, so here we see Professor John van der Oost. And he is, as we in Wageningen would like to think, he's the godfather of CRISPR. So he was the one that invented or found that CRISPR is a bacterial system that provides resistance, or let's say that can prevent our virus that basically attacks virus particles. And John van der Oost, he was in the Dutch newspaper on last year. And basically, I will translate it for you here. He says, we are currently building a CRISPR enzyme to optimize it for use in plants, a new CRISPR enzyme to provide a novel or a better resistance against drought, heat, and certain plant diseases in, for example, crops like rice. And we are planning to provide these patents for these technologies for free to especially developing countries. And basically, this is what the CRISPR for all movement is about. So, what we would like to do with in CRISPR for all is really to provide a level playing field for all breeders and to provide accessible access and practical experience with genome editing. And this movement was launched at Crop IB in Wageningen this spring. And it is really a EU-rooted genome editing platform with success in three crops so far. It's built on the thermal-Cas9 enzyme. I will tell a little bit more about that later, as it has some unique properties that I believe makes it very interesting indeed. So the difference in what CRISPR-Val would like to offer is really to have the innovation, so the CRISPR system, accessible, to provide it immediately, and also it should be very affordable. And that is then tailored to the size of the company. Depending on the size of the company, you have a different fee structure. And the fee structure is also an annual fee. And there are no reach-through clauses. So also part of your profit margin, if you're a smaller company, will not go to the CRISPR for all movement. Therefore, we think it is also more affordable for smaller and medium sized businesses. This innovation has been protected by a number of patents, and at Wageningen we did a patent analysis, but to also see if other patent councils agree with our analysis, we did it with an independent analysis done by NLO. And based on those analyses, we had the confidence to continue with the CRISPR for all movement, as it seems that we are able to provide this aside from the currently available CRISPR systems. So it is really Wageningen-rooted technology. Mainly it comes from the group of John van der Oost, so from Wageningen Microbiology. And what they do, they have optimized this enzyme via specific ways of enzyme engineering. And then what initially happened is that we at Wageningen Plant Breeding, where I'm working, we would obtain this enzyme. We would then optimize it by providing different promoters and terminators and optimizing it, codon optimizing it for higher performance in different crops. Then we test it, and there's a feedback loop that has been ongoing for some years. So it's really a multidisciplinary work that we are doing. We also quite some traction. So we got a Dutch grant from 1.9 million. And also we have Gates One and an F Foundation that support us. And that's how also we got plant physiology and molecular biology involved, including also the IGI that work together on optimizing and testing the enzyme in different crop systems. Then what I would like to do now is to switch gears a little bit and talk about, okay, what exactly is the CRISPR technology that we offer? So it comes from thermal-Cas9, which was found close to Wageningen in the Netherlands in a large steamy compost heap in which we found a thermophilic bacterium. And within that bacterium, there was a different Cas enzyme, thermal-Cas9, that was active at 60 or 50 to 60 degrees Celsius. And because of this high activity, it's a very robust enzyme. And also, it is a completely different sequence identity compared to, for example, SP-Cas9, less than 25% nucleotide sequence identity. So we were enthusiastic about this enzyme, and the idea was to test it in plants to see if we could indeed use it there. But initially, we saw that in planta there was almost no activity at all, so it was not usable. And the cause was that indeed it is active at these very high temperatures of 50 to 60 degrees Celsius. However, not at the temperatures where plants thrive, and therefore we did not see a good editing efficacy there. We did not give up, and we started the optimization of the protein, especially done by the group of John van der Oost, so the microbiology group in Wageningen. And there were a number of ways in which we did protein optimization. including some error-prone PCR systems, AI methods, and using AlphaFold to cleverly try to change a few nucleotides to destabilize the protein and therefore have it active at a lower temperature. So initial results showed editing in rice and also in stably transformed potato plants and potato protoplasts, which I will show some data from in a bit. But first, maybe to show the differences between SP-Cas9, so the conventional CRISPR system, and our thermo-Cas9. So the origin is, as you see, different. So it's not from a Streptococcus, but from a Geobacillus. The cutting type is similar, highly similar even. So it's thermo-Cas9 is a blunt cutter, just like SP-Cas9, and it cuts 3 base pairs upstream of the PAM sequence. Regarding the PAM sequence, it is not NGG. It is an 8 base pair PAM sequence, which looks a bit daunting, perhaps. But when I am designing guides for thermo-Cas9, it is not that much more difficult to find suitable guide RNAs for the thermo-Cas9 system. And then finally, regarding the enzyme size, we see that it is roughly 10% smaller than the conventional CRISPR system SP-Cas9, which might be nice for RNP-based delivery systems. So another big difference is that thermo-Cas9 activity is temperature dependent. So you can activate it and inactivate it by changing the temperature, but I will show a little bit more about that later. So then switching to the initial results from 2023 in rice. This is in protoplasts. So we have a transient system where not all the protoplasts are transfected. And what a protoplast is, maybe for some of the background, it is a plant cell from which we remove the cell wall. And then if you use this process, you have hundreds of thousands of cells that you can transfect with your CRISPR thermo-Cas9 or SP-Cas9 construct, and then incubate it for 48 hours and see, okay, what kind of editing efficacies did we find? So did we find mutations at the target site? We tested this in SP-Cas9 for a Werke 53 and MPK2. And then we saw initially that SP-Cas9 indeed did outperform our thermo-Cas9 enzyme with around 40 to 61 lower mutation frequency in thermo-Cas9 compared to SP-Cas9. We did further optimizations and also, actually, I should definitely mention that this was work done by Krista Testring's group, and special thanks to William and Joram. And William is the one that did these analyses here. So then switching to tomato, or rather potato, in protoplasts, we also did testing. That was done by Jan Schaart from the plant breeding department. And we again compared thermo-Cas9 to SP-Cas9 with the same target site. And then we could see that thermo-Cas9 was back then operating at 25% the efficiency of SP-Cas9 in these protoplasts again. We also did the same thing. We tested in stably transformed potato plants, and here we could see the same trend, let's say. Further optimization was done, and last year we found that finally we have also a really competitive editing with thermo-Cas9 compared to SP-Cas9. And this was done in tomato then. So again, we see the editing efficiency on the y-axis, and we see SP-Cas9 in yellow on the left. And I used a guide RNA or CRISPR SP-Cas9 system for my PhD, which I knew was very highly efficient. And around that target site of SP-Cas9, I generated 10 randomly chosen thermo-Cas9 guide RNAs. And so I did protoplast transfections, and then we can see that depending on the guide RNA that you choose, in absolute sense of editing, the guide RNA can, the thermo-Cas9 system can outperform SP-Cas9. We tested this not at one locus, but at 2 loci, and we saw a similar trend in the other locus as well. So the next thing that I would like to mention, and I quickly look at the time. We have some novel applications that thermo-Cas9 is interesting for. So I think the one that is most interesting is turning it on and off by changing the ambient temperature. So first of all, a use case for this could be that you grow a plant and you transform it stably with your CRISPR thermo-Cas9 construct, and you keep the plant at a low temperature. What happens then is the plant will be transformed and it will generate inactive thermocas9 enzymes because at the lower temperature it does not cut. However, the enzyme does accumulate over time. So this is yeah, then you get a higher concentration of thermocas9. What you can then do is place your stably transformed plant at a higher temperature. Let's say you move it from a lower temperature or below 18 degrees to, for example, 30 degrees. And what happens then is that thermocas9 rapidly becomes highly active. And this can have many advantages. For example, if you want to grow your plant initially with a where at a stage where the enzyme should not be active, and you want only at a specific stage of the plant or of the plant you want editing, for example, during the flowering stage, you will get edits only then. Or if you want to reduce off-target effects, you can activate and inactivate the system. If you want, you can also inactivate the system later and repeat the heat treatment if desired. Um, Briefly then about another use case of this heat shock. So of activating it and inactivating it, it might be possible to increase the rate of targeted crossovers. So let's say you have your heterozygous plant, your tomato plant, with two homologous chromosomes, and you use CRISPR to target both homologous chromosomes. What happens if the cuts are simultaneously occurring? You will get four broken DNA ends that the plant cell will try to repair. And if the repair happens where the one chromosome fuses to the complementary other chromosome, you will get a targeted crossover, which using SPCas9 is a very rare event because you cannot steer when CRISPR-Cas is active. And therefore, it cuts occasionally in one chromosome and occasionally in the other. But if you can steer when activity occurs, for example, with thermocas9, you can cut both homologous chromosomes at the same time and thereby increase the rate of these target sites being available and increase the rate of target crossover. Um, I have an example here, but I think for now, judging the time, I think I will skip this one and move on to broadening validation that we're currently doing. So as I mentioned, we have a increased size of the consortium, and we are focusing now not only on tomato, potato, and rice, but also on maize, barley, and sorghum. And we are extending this as we yeah during these phases of the project. Um, as I mentioned, it is not like a one-man job. This is a quite big team that is behind it. So I would like to thank all the people that are here, including the people from the IGI, and the Gates, the Novo Nordisk Foundation, and the Biotech Booster projects. And to conclude, maybe I would like to show also the licenses that we are currently already offering. So we have a test license where you can, as a breeding company, test whether you get good results with thermocas9. If you do like the system, then you can get a research license. And if you want to provide or make a commercial product, then we can also offer a commercial license. So with that, I would like to finalize and finish the presentation. And thank you so much for your attention.
Thank you very much. That's super interesting and really fascinating to have this tool potentially available. Thank you very much, Jillis, for introducing the CRISPR for All project. Great that you have so much funding. 7 million euros from the Novo Nordisk. That's very nice. Very nice indeed. Okay, so any questions, please put them into the chat. We've got an initial question about RMPs from Casey. So he suggested different smaller enzyme, but in general, do you have evidence that using a smaller Cas enzyme will change the efficiency if you use RMPs?
So we are currently testing this, and we would like to continue this. With the initial results, we see that indeed this is the case, but we don't have a hard data on this.
Okay. So you mentioned that the thermo switch goes from 18 to 30 degrees. So have you tested a range of temperatures? Do you see more editing if you go even higher, and then you run the risk of damaging the plants, I guess?
Yeah, this is indeed something that we're currently exploring. So what we see is that at a higher temperature, the enzyme is more active. But also, if you would prolong the heat shock, for example, in potato, the genotypes that we are using, they are not that tolerant to these higher temperatures. So then you get a decrease in cell viability there.
Okay.
So indeed, it depends also on the enzyme that the thermocast9 enzyme that we use because we are continuously optimizing it.
Mhm.
And then some of them they are active at a higher temperature compared to others. So in the end, we would ideally have a range of or a number of different thermocast9 enzymes that could be crop specific.
Mhm. Okay. So a question from Pascal. Good to see that efficient alternatives are coming. Yes, indeed. So have you noticed any change in off-target effects using Thermo-Cas9 compared to
So far, we have not looked too much into the off-target effects. Something that we are planning to do, but currently we don't see differences per se in Thermocas9 and SPCas9 in off-targets because we did some initial tests, but that is we don't have the hard data on this yet.
Okay. Question from Andreas. Have you regenerated the edited potato protoplasts after the editing yet?
For the potato protoplast, no, we did not focus on regenerating protoplasts from calli. So these were the two different experiments where in one we did protoplast transfection, and then we just tested the protoplast by PCR-based amplification of the isolated DNA, and then analyzing it with CRISPR-Cas2 as our tool for indel analysis. And in the other case, we stably transformed potato plants and did a similar approach. And yeah, so it was not regeneration from protoplasts.
Okay. Question from Gertz. Do you use Thermocast already for base or prime editing?
Um, Currently, we get this question more often. Currently, we're not. We have initial work done on it, but we're not currently exploring this further optimization. So this is something that is definitely on our wish list.
Okay. Okay. So I have a just before we wrap up a general question about how this was discovered. So I guess there was an experiment looking some piles of manure or something close to the university, and they found this bacteria. And you
Yeah, exactly. So the group of John van Oost is the microbiology group, and he was interested in testing or looking at different bacteria, and then this thermo-tolerant bacterium came into the picture, and yeah, from there it was yeah the thermocas9 enzyme was identified. And after some years of research, they got into contact with the plant breeding group, and we together decided, okay, let's test this in plants.
Mhm.
Uh, and so it was more from a very enthusiastic approach and standpoint to see, okay, can we use this system also in plants? Initially not, but via optimization, indeed, we.
And do they have other candidates that they identified from their initial screens?
So I think I know that in Wageningen there has been some studies where they focus on looking at the novel Cas enzymes, but I don't think they have been so successful compared to the thermocas9 experiments that we have been doing.
Great. So interesting to think that given the amount of bacterial strains there are, bacterial species in the world, that they're all going to have their Cas enzyme of some type, that the usefulness of them out there. So thank
Mhm.
you very
Yeah.
much, Jillis, for giving the talk today. It's been really great to hear that.
Thanks so
Um,
much for having me. Yeah.
it's I should, as Good says in the chat, just to remind everyone, we have another PlantEd webinar which is scheduled for September. So as you joined this one, you'll be on our mailing list to promote that as well. So we will have another webinar in September. So for now, though, let me thank Claudia and Gillis, and I hope everyone has a nice day, nice summer, and we will see you in the next PlantEd webinar at the end of the summer. So thanks very much, everyone, for attending.